So, Michael, you and I talked earlier about how there are different considerations when sourcing autologous stem cells. So, I'm curious, in your experience, what unique challenges need to be considered when using a patient's own stem cells as a starting material for cell therapy?
Yeah, I mean, it's a good question. It's something that we've talked about for years, right? And so when sourcing a patient's own cells for starting material, you know, that end-to-end journey from patient to manufacturer, all along that journey there could be challenges, whether it's labeling or changes in scheduling, yielding and actually manufacturing product for patients. So it really depends on what part of the journey those challenges could happen. But you know, the starting material is the key that you need to manufacture patient cells, and again, getting them to and from the patient logistically from that supply chain perspective, all kinds of challenges and hiccups that occur in that end-to-end process. And we can get into some of those specifics a bit later, I think.
Yeah, no, I'm excited to dive into the supply chain logistics a little later on. I'll pose this question to all three of you before we move on here. In terms of autologous stem cells, from a manufacturing point of view, as the raw material, what are the bigger challenges that may arise? Whoever wants to answer that, feel free.
I think as we think about it, right, you know, there's a lot of focus on commercializing these products. So for me, when I think about it, the first thing I think is, how can you scale this, right? Most companies can do this for 6 to 8 patients per month, maybe 10 to 12. You know, you slowly go through that process when you have clinically efficacious products. But realistically, can you commercialize this, and can you scale smoothly, and can you actually have an orchestrated platform where patients and customers alike can have a good experience from, you know, cell collection to infusion? And it's a tough task. It's really hard to get that smooth process. But, you know, as we go in, the further we go here, the more products that become commercialized, it really is time to I think do better at scaling these operations and scaling products so that realistically this is a commercially successful endeavor when we think about the operations and logistical aspects of these products at scale.
Thank you, Michael.
Yeah,
sorry,
Tyler, I can jump in on that one as well. And of course, this is, as I said before, I'm really developing allogeneic cell therapies, but as a physician scientist, you know, I still see patients on the transplant service. And I think when I originally started this work, and this is not at all a dig towards autologous therapies, right? Because I think autologous therapies have really changed. They've been game changing, right? The fact that we have approved therapies that cure patients with leukemias and lymphomas is without a doubt is game changing. I think when I started this work in the early 2000s, and the first trial we started in 2006 was a patient-specific trial, meaning we would identify one cord blood unit that was specifically for certain patients. So like autologous, you know, I could only use those cells for that patient, right? And I think we quickly realized that this is really hard to do, really stressful for the poor people who are manufacturing, right? And like the techs I had in the GMP facility, what happens if it didn't work out, if they dropped the flask of cells or whatever. And we quickly wanted to try to figure out a way to pivot this. And I think most of us would agree that allogeneic offers the potential to overcome the deficiencies of autologous, but we still have a long way to go. And I think with autologous therapies, one of the things that has always concerned me is when you are taking cells from a patient who has been heavily pretreated, we don't understand the quality of that starting material. We can't define it, right, until you sort of harvest that patient and get those cells and see what's there. But you don't always know that the material you're getting is going to be healthy enough to actually generate that final product that you want to generate. And so we really wanted to think about, how do we always have qualified starting material? How do we know what we're starting with and really know what we're ending with? And again, this is not, autologous therapies have been game changing, but they are expensive. They are I'm sure Michael will say, they are logistically incredibly complicated. It's one manufacturing run for one patient. Every patient is different. So your donor to donor variability is incredibly high. And so we kind of came at it from that perspective, right? this is our starting point. We knew we were not going to be developing products that would live forever, and potentially there's safety along that as well, right? We knew in the allogeneic space, our biggest risk is rejection, right? And that we will generate a product that won't live long enough to do what it needs to do. But I think we will get there. So for the autologous thing, one of the big concerns for me is you don't know what you've got to start with, especially in a patient who's been heavily pretreated, and you don't know you're going to have a product at the end.
Yeah, I would agree with that, and my personal concern on autologous is really around cost of goods and how that correlates to patient access, and that's really why I've joined BitBio a couple of years ago. But the dream for allogeneic cell therapies, of course, is to kind of go learn from what we've learned from autologous cell therapies, apply that to allogeneic, and be able to lower the cost of goods, orders of magnitude, so that anyone could be able to access the therapy. It becomes medicine rather than therapy. But there will always be a place for autologous for certain indications. And so I think that if we could focus that area on reducing the cost of each run, that would be fantastic.